Hello everyone. In this video, we are going to see respiratory balance sheet. We will see how many ATP molecules are obtained from single molecule of glucose that has undergone oxidation during aerobic respiration. During aerobic respiration, all the 6 carbon present in the glucose undergoes oxidation and gets converted into carbon dioxide molecules. 2 were released from the second step link reaction and 4 gets released from the third step citric acid cycle. The energy that was present in the covalent bonds of glucose gets trapped or extracted by extracting electrons or hydrogen during this oxidation process. These electrons or hydrogens are then transferred to coenzymes, resulting in the formation of reduced coenzymes. Totally 12 reduced coenzymes were formed from the first 3 steps of aerobic respiration. Of the 12, 2 are NADH molecules from the first step glycolysis, and 2 again NADH from the second step link reaction. In the third step, 6 NADH molecules and 2 FADH2 molecules were formed. Now these reduced coenzymes, when they transfer these electrons to electron transport chain, the energy that was extracted during this transfer was utilized to synthesize ATP molecules by the process oxidative phosphorylation. The molecule that was involved in this process was ATP synthase. So totally 32 to 34 ATP molecules were formed by oxidative phosphorylation. Apart from that, by substrate level phosphorylation, 4 ATP molecules were synthesized, 2 from glycolysis and 2 from citric acid cycle. When one NADH molecule transfers pair of electrons into fourth step of aerobic respiration, 3 ATP molecules will be formed by oxidative phosphorylation. Similarly, if one FADH2 molecule hand over pair of electrons into electron transport chain, 2 ATP molecules will be formed by oxidative phosphorylation. Now let's see this table. From the first step glycolysis, 2 molecules of ATP are synthesized directly by substrate level phosphorylation and 6 ATP molecules are synthesized from 2 NADH molecules that are produced during this process. So totally net gain is 8 ATP molecules from glycolysis. From the second step link reaction, 2 molecules of NADH were produced. These molecules, when they hand over the electrons to electron transport chain, they could result in production of 6 ATP molecules by oxidative phosphorylation. In the third step citric acid cycle, from 6 NADH molecules, totally 18 ATP molecules can be synthesized by oxidative phosphorylation. Similarly, from 2 FADH2 molecules, 4 ATP molecules can be synthesized by oxidative phosphorylation. Apart from oxidative phosphorylation, 2 molecules of ATP are synthesized by substrate level phosphorylation in citric acid cycle. So if you count these numbers, you will get 38 ATP molecules, but in some textbook, you will get the range as 36 to 38. Already we have seen the reason for this difference. The 2 NADH molecules that were produced during glycolysis were actually produced in the cytoplasm. These NADH molecule cannot directly enter into mitochondria to hand over the electrons into electron transport chain. Based on which shuttle system is involved in transferring those electrons from NADH molecules, accordingly, this number will also vary. Based on the shuttle system, electrons from these NADH molecules are transferred either to NAD+ or to FAD. If they are transferred to NAD+, in such case 6 ATP molecules are obtained, so total number will be 38 ATP molecules. But if the electrons are transferred to FAD within mitochondria, in such cases only 4 ATP molecules will be obtained. So there will be production of only 36 ATP molecules per single molecule of glucose. Whatever calculations we have seen so far is based on certain assumptions only. We assume all pathways occurs sequentially and orderly. Electrons from NADH is transferred into mitochondria. None of the intermediate in the pathway is utilized to synthesize any other compound. We also assume only glucose is respired and no other respiratory substrate is entering into the pathway at any of the intermediary stages. But these kind of assumptions are not really valid in living system. In living cells, all pathways occur simultaneously. Substances enter and are withdrawn from pathways whenever necessary. ATP is utilized as and when needed, and enzymatic rates are controlled by multiple means. Then the question will be why we need to do this calculation. By doing such calculations, we can easily appreciate how beautifully and efficiently living systems extract energy from biomolecules and store them as ATP molecules, that is, energy currency for different functions. From these calculations, we can also understand that there can be a net gain of 38 ATP molecules during aerobic respiration of 1 molecule of glucose. So far, we have seen how much of energy is obtained from 1 molecule of glucose during aerobic respiration. Already we have seen what happens to glucose when it enters into fermentation. In the next class, we will see what are the major differences between fermentation and aerobic respiration.